I'm David Cole, the President and CEO of the Science History Institute. We are delighted to welcome you to the 31st Annual Ullyot Public Affairs Lecture featuring Dr. Peter Agre. This annual award and lecture, endowed by chemist Glenn Ullyot, seeks to illustrate how chemistry, biology, and the sciences in general contribute to the public welfare. Glenn once said that, quote, "Chemistry, biology, and physics are the basic sciences that are key to understanding the world around us. It is our mission that each Ullyot lecturer will stimulate more people to appreciate the positive impact that these sciences and the researchers who pursue them have on our daily lives," unquote. I believe Glenn would be pleased with this annual lecture series, as it continues to bring people of remarkable scientific accomplishment to the attention of the general public. Among our past Ullyot lecturers are Robert Langer, Phil Sharp, Jennifer Doudna, and Roald Hoffman. And now, Dr. Peter Agre has honored us by joining this select company. The Ullyot Lecture is particularly important to the Science History Institute, as it enables innovative figures in science and medicine to bring their stories to an ever-growing global audience. These are stories of groundbreaking scientific achievements that furthered scientific inquiry while also leading to improvements in our quality of life. In a time where our world has been visibly altered by the effects of both the pandemic and climate change, we welcome the words of those who possess unique and deep understandings of the scientific foundations of challenging phenomena. I am delighted to say that the Ullyot Public Affairs Lecture is jointly sponsored by the Science History Institute, the Department of Chemistry at the University of Pennsylvania, the Department of Chemistry and Biochemistry at the University of the Sciences, and the Philadelphia section and Delaware section of the American Chemical Society. Together, we and our sponsors are pleased to bring you the 31st Ullyot Public Affairs Lecture. I should note that tonight's presentation is the first virtual Ullyot lecture, but in keeping with tradition, there will be a question and answer period after the lecture. Please enter your questions in the chat, the email, or social media as the program progresses. And Charlotte Abney Solomon, the Science History Institute's Associate Director of our Beckman Center for the History of Chemistry, will moderate. And now a word about our distinguished lecturer. This evening, Nobel laureate and medical scientist Peter Agre will discuss the special role of water in biology and his transition into malaria research. His talk is entitled Aquaporin Water Channels, From Atomic Structure to Malaria. Dr. Agre is Bloomberg Distinguished Professor and Director of the Malaria Research Institute at Johns Hopkins University. His love of biomedical research began while working on cholera as a student in the laboratories at Johns Hopkins School of Medicine. After his residency and a fellowship, he returned to Johns Hopkins, eventually joining the School of Medicine faculty in 1984, where he rose to the rank of Professor of Biological Chemistry and Medicine. He is a member of the National Academy of Sciences and the Institute of Medicine, for which he has chaired the Committee on Human Rights. From 2009 to 2011, Dr. Agre served as president and chair of the Board of Advisors of the American Association for the Advancement of Science. He has also been elected to the American Academy of Arts and Sciences and the American Philosophical Society. In addition to winning the Nobel Prize in Chemistry in 2003 for the discovery of aquaporin water channels, he has received the 1999 Homer Smith Award from the American Society of Nephrology and the 2005 Karl Landsteiner Award from the American Association of Blood Banks. He has 16 honorary doctorates from universities around the world. Dr. Agre received a BA in chemistry from Augsburg College in Minneapolis and an MD from Johns Hopkins School of Medicine. He completed his residency at Case Western Reserve University in Cleveland and an oncology fellowship at the University of North Carolina, Chapel Hill. While best known for the discovery of aquaporin water channels, Dr. Agre is a gifted storyteller who has been known to actually sing the periodic table to his students. So in addition to being a brilliant scientist, He clearly has a great singing voice. And now it is my great honor to present our 2021 Ullyot Public Affairs Lecturer, Dr. Peter Agre.
It's my pleasure to be the Ullyot lecturer. Thank you, President Cole, for the introduction. Thanks to the sponsors, and thank you all for tuning in. This presentation is going to be a summary of our aquaporin work. I'm going to present a few chemical details, but try to keep things on a level that will explain who did the work. Science is done by young people, and we had a wonderful group of young people in our laboratory over the years. So water, it's in the news all the time. Global climate change, melting of the Greenland ice cap. Certainly global water issues are very important, but water is also important in biology. Water is the major substance in all of the cells of all the tissues of our body and of other life forms. So water is really the essence of life. But water disequilibrium can be quite dangerous. This tranquil Arctic river my son and I paddled a few years ago became a very challenging rapids. So water is both essential but has potential dangers. Usually scientists are pursuing a hypothesis. The discovery of the water channels, the Aquaporins as we now call them, was a sort of an example of reverse science. It was a serendipitous observation. And our laboratory, I'm a trained hematologist, was studying the rhesus blood group antigen. The Rh antigen, of course, is very important in red cells. It's important for problems of maternal fetal incompatibility, but it had never been isolated and characterized. So we set about doing that and proceeded in our isolations of the Rh polypeptide to bring in a second polypeptide, a slightly smaller size of 28 kilodaltons, which we thought was a proteolytic fragment of the Rh, but in fact it turns out it was a total unrelated protein, a contaminant. But it was figured out first by Brad Denker when he was a medical resident in our laboratory before he joined the Harvard Medical School, where he's now a professor. This 28 kilodalton protein had some features that we found very interesting. It didn't stain with the usual protein stains. This is a silver stain, which is a very sensitive form of stain. And it turned out that it was a very ubiquitous protein and had never been isolated or identified even in red cells. So it caught our interest, but really not because we had some great hypothesis. But it turned out to be easy to purify. We were able to determine an amino acid sequence of the amino terminus and clone out the complementary DNA, thinking that this would tell us what the protein is and what it does. And the deduced topology of the expressed protein shown in the diagram on the right indicates that it looks like it's a bilayer spanning protein, snaking through the membrane 3 times, and then, interestingly, going through 3 more times. And the first half of the protein and the second half of the protein are genetically related, not identical, but the loops B and E, which contain a perfect canonical sequence, which turned out to be quite important. But when we went to the genetics database, now this is in the early 1990s, the human genome had not been sequenced. We found several interesting relationships. Proteins from the lens of eye, proteins from brain of insects, bacteria, and plants. But there was no functional definition of what any of these proteins did. So we were in a situation where we had a new protein, we had the DNA, but we didn't have really a clue what it did. So this is where science becomes very challenging. You can talk to your colleagues, but if the colleagues don't have ideas, you can't find something in the library, Where do you get your inspiration? And I think it's from talking to the scientists in your university and at other universities. The answer for how we got onto the track that this protein is a water channel came from a family camping trip. Shown here are my 4 kids. Mary and I are very proud of them. This is when they were small and cute. Now they're grown. The little girl in the front with the curly hair, Carly, is now a healthcare social worker in Philadelphia. They've gone on to their careers, but when they were little, we would camp at the national parks every year, and they loved it. After doing this for a few years, Mary said, let's ask the children which national park they'd like to go to next year. And the kids all yelled Disney World, which is, of course, not a national park. We went to the Everglades, we went to Disney World, and on the way back to Baltimore, we stopped in Chapel Hill, North Carolina, where I shared the story of this new protein that puzzled us. So abundant with homologs in a variety of life forms, what could it be doing? And it was a conversation with John Parker. John was a professor of hematology at UNC Chapel Hill. And it was John who first put it all together. He was a very good physiologist. He said, Peter, this protein is in red cells, renal tubules, plant tissues. You realize, of course, that these are all highly water permeable tissues, uniquely so. Well, I didn't realize that, but his suggestion was very, very moving. And so when we returned to Johns Hopkins, I teamed up with Bill Gugino to clone out, to express the protein. John also went on to explain that water channel would be a wonderful identity because physiologists have been searching for the explanation for how water crosses cell membranes for more than a century. Charles Overton, an early physiologist, looked at the problem of cell membranes and realized that lipophilic molecules, such as gases and alcohol, would pass through the lipid part of the membrane, but hydrophilic molecules, sugars and ions, would need to go through protein pores. This is oftentimes referred to as Overton's rule. And it also reflects the basic chemistry of water. We were taught, of course, as children in our science classes, water is H2O. But in gaseous form, the H2O molecules are free from one another. In crystalline form, in snowflakes, they're all organized perfectly, but in fluid, in bulk solution, hydrogen bonding between adjacent water molecules will allow protons to hop from one molecule to another. It's important that the water channel be permeable to water, but not protons. So now we have a view of the membrane permeability. We realize that all lipid membrane bilayers have a small degree of water permeability by simple diffusion. But the putative water channels have a name now. We call them the Aquaporin water channels, and that's the official name for this family of channels. And these permit very large amounts of water to cross the membrane. Again, they're expressed in some tissues, not all tissues, and they're highly selective. Water flows, but protons do not. The movement of water follows the osmotic gradient, and there are inhibitors and a low Arrhenius activation energy. So the expression of the protein was the essential step. And we were fortunate to team with Bill Gugino, our colleague at Johns Hopkins. Bill is an expert physiologist and had the Xenophus laevis oocyte system in his laboratory. Frog eggs. So the test that he would do is the cystic fibrosis transmembrane regulator, but we looked for simple osmotic swelling. The oocyte on the left and the real oocyte is about 1 millimeter in diameter. The oocyte on the left is injected with buffer alone, and the one on the right was injected with 2 nanograms of the complementary RNA for this new protein. They're expressed in culture, and there's no obvious difference until we drop them into distilled water. And as we hoped, the test oocyte swelled rapidly like popcorn and exploded, producing much jubilation in the laboratory. This is a common feature that any scientist will recognize. And this is Greg Preston, the wonderful postdoc who did many of these early studies. And I have to tell you the truth, Greg is still celebrating 2 years after the first paper when I took this photograph. He's a wonderful young man. He's now at a biopharmaceutical company in Boston. Verification of the function was feasible because we had purified the protein. And when membrane lipids are reconstituted, they will form membrane vesicles. This EM fracture replica shows smooth-walled unicellular vesicles. When the lipid plus the pure protein are reconstituted, in the lower panel, you see that the smooth membranes now have little bumps, each bump representing a reincorporated water channel protein. And we tested for the ability to shrink. The difference between the permeabilities is quite clear. Simple lipids have a finite degree of water permeability, but it's pretty rapid. It reaches equilibrium at about a half a second. In the lower tracing, you can see with the presence of the aquaporin protein, the permeability is very fast and reaches equilibrium at about 10 to 20 milliseconds. So it's an increase in velocity of water movement. Now, as a biologist, we always want to know the mechanism of function. And for this, we were fortunate to team up with two outstanding investigators, Yoshinori Fujiyoshi in Kyoto and his team, and Andreas Engel at the University of Basel in Switzerland and his team, including a wonderful graduate student, Tom Waltz. Tom is now a professor at Rockefeller. So over a course of a few years, we were able to, using the cryo-electron microscopy, to solve a high-resolution structure of the aquaporin 1 protein. And on the left panel, you can see the view from the top of the channel, looking down through the cell membrane. That hole in the center of the donut is about 3 angstroms in diameter. That's just larger than the size of an average water molecule. And on the right panel, we have a cutaway. You can see the pore connecting the top from the bottom. There's a little bit of side-to-side movement, but it's basically a shear tunnel permeated by water. And to be able to pass water, but not pass protons, there has to be a block somewhere in the channel. And the structure that I just described, we refer to as the hourglass, because the 2 repeat loops bend in from the outside and the inside of the cell membrane into the center of the bilayer, where they overlap. And so if you look at the panel at the top, you see the pathway of water through the channel, and in the very center, there are 2 highly conserved residues in the 2 repeats, which separate water molecules for a billionth of a second, preventing the movement of protons. This is important because, as we'll see shortly, these proteins permit us to reabsorb 99% of our glomerular filtrate every day. Our kidneys will release almost 50 gallons, 200 liters of water into the primary urine. And of course, we would not survive if we released that volume of water. 99% of it is reabsorbed. But at the same time, we have to be able to release acid loads. So the water passes through the cell membrane, but in allowing the urine to be concentrated, but the
So I hope this now is, are we okay, Alexis?
Okay, I think we are good to go and you are all set. So I'm going to let you return to your fantastic talk. Bye, everyone. Thanks for your patience.
I have to admit, I think it was a lot easier to win the Nobel Prize than to work on my computer. So I'm talking briefly about the presence of the aquaporin 1 protein in the kidney, where it's in the proximal convoluted tubules, and allows us to concentrate our primary urine. Again, we release almost 200 liters of primary urine from our plasma each day, and we must reabsorb the water. And it enters through the epithelia from the apical surface, through the cytoplasm, and exits from the lateral basal membranes, following a small standing osmotic gradients created by the vectorial separation of salt and sugar transporters. So as a medical doctor, of course, we're always looking for information that will help us improve health. And this new gene, aquaporin 1, was localized to the short arm of human chromosome 7, where it overlay the general site where the Colton blood group antigens had been localized. And it seemed quite clear, since there are about only 25 families of blood group antigens, that our protein and the blood group antigen were the same molecule. And this is work done by Chul So Moon, a graduate student from Seoul, Korea. In fact, that turned out to be correct, because in the whole world, there are 6 null kindreds having no Colton A or B. And we were able to get their DNA. We found the polymorphism at an extracellular site, a very subtle difference, an alanine-valine polymorphism. But then we turned our attention to the null individuals and found that they contained knockout mutations. They were homozygous for knockout mutations. They could not make the aquaporin 1 protein in any tissue. We thought this would probably be of severe clinical significance, but it turned out the individuals felt well and they looked well, and shown with permission in the center of this photograph is an aquaporin 1 null individual. She can't make the protein, but she was identified because when she attempted to donate blood later in life, she had high levels of antibodies, because when she was pregnant, she became sensitized. And so the young man in this photograph, Landon King, who was at that time a postdoc, he's now the executive vice dean of Johns Hopkins Medicine. He did the phenotyping and found that, lo and behold, these individuals have very normal renal function, except they cannot fully concentrate. So on an average night, when we get 7 or 8 hours of sleep, we're fluid deprived, and we concentrate our urine up to about 1,000 milliosmolar. These individuals can only concentrate up to about 400 or 420. So they're at no risk when they have access to free water, but they would be at a grave disadvantage if they were in a desert or in a nursing home, unable to ask for water. The interest in the aquaporins was really amazing. Many groups got involved, and it turns out there are many different aquaporins. The human repertoire includes 13, of which most have been well characterized. And they are either permeated by water alone, which we refer to as classical aquaporins, or permeated by water plus glycerol, and we refer to those as aquaglyceroporins. And I'll tell you a little bit about some of them. The collecting duct, as I mentioned, where vasopressin works, is a site where aquaporin 2 was identified. It was cloned out by a group in Japan and characterized by our colleagues at the National Institutes of Health, Søren Nielsen, together with Mark Knepper and other investigators, also pursued this. It functions very differently from the aquaporin 1, because the aquaporin 2 normally resides in the collecting duct, the distal part of the kidney, and it resides in intracellular vesicles. But vasopressin, referred to as AVP in this diagram, will activate receptor and lead to exocytosis of the intracellular vesicles. And at the far right, you can see water can now enter the top of the cell and exit from the bottom, passing through the collecting ducts. This may seem rather esoteric, but it's very important in our daily lives. Vasopressin is the final concentrator of our urine, and it makes a big difference. If you have a full bladder and you're in the economy section of an airliner, you know how urgent that problem can be. Clinically, they're also quite important. Inherited defects are rare, but common defects are found in all types of fluid retention, including congestive heart failure, pregnancy, and underexpression is found in all forms of enuresis, including bedwetting by small children. Fortunately, the kids outgrow it. Another member of the family, we refer to as aquaporin 0, is expressed in lens. This is work of Masato Yasui, a postdoc in our lab who's now back in Japan and chairman of the pharmacology at Keio University. So mutations cause cataracts. Cataracts can be cured surgically. Yet another member of the family, we refer to as aquaporin 4, the water channel in brain, turns out to be very pivotal in terms of brain homeostasis. And our colleague in these studies was led by Ole Petter Ottersen, a neuroscientist from Norway who's presently the rector of Karolinska Institute in Stockholm. In brain, capillaries are surrounded by a basement membrane, And they are surrounded by astroglial end feet. The diagram on the left, borrowed from the textbook of Eric Kandel, shows that the astroglial end feet, they look like suction cups, will align around capillary, and just below the basement membrane, the black dots represent specific staining. So this is the barrier where water is trapped, the so-called blood-brain barrier. John Neely, who was a graduate student in the laboratory, noticed some key motifs within the structure. John's now a neurologist in Boston, but his work was pivotal and showed that the distribution of the aquaporins in normal brain, this is now a rat or mouse brain preparation, are clustered along the basement membrane. In the syntrophin null mice, these are mice that have been targeted for disruption of the syntrophin, which is a bracket protein which holds the aquaporin 4 at the basement membrane. The protein is present, but it's at the wrong membrane. And these mice were developed by Stan Freiner from the University of Washington and Marv Adams. Mahmoud Amiri and Ullyot Petter's group did a pivotal series of studies and found that the aquaporin 4 in normal mice serves as a hazard after a defining brain injury. Mutant mice, shown on the right, have a preservation of brain tissue after occlusion of the temporal artery and reperfusion. This suggests that inhibitors of the aquaporin 4 might be useful after head injury to prevent brain swelling, because brain edema oftentimes is the fatal consequence of a head injury, a stroke, or in a brain tumor. Shown here is an MRI of an individual with a frontal lobe meningioma, a benign tumor, but the location, of course, is extremely hazardous. The MRI, when weighted for the density of water, you can see in the upper right panel, shows massive edema in the frontal lobes. Fortunately, the patient had the tumor removed and the edema dissipated, but it took several weeks. So the outcome was great, but you can see the consequences of edema can be very hazardous. Yet another member of the aquaporin family, aquaporin 5, expressed in salivary glands, lacrimal glands, and sweat glands, cloned out and studied by Surabhi Raina, a postdoc from University of New Delhi, who's now a professor up at Syracuse University. So aquaporin 5 is the last membrane water crosses during the generation of these fluids. In mice who have aquaporin 5 knockouts prepared by Carissa Crane and Anil Menon at the University of Cincinnati, they have sweat glands, but they're hypofunctional. These mice cannot really sweat. And of course, if they were living in hot environments rather than cool, dark environments, they would be at risk. And of course, humans work in the very dangerous hot temperatures. The ability to sweat is essential for survival. The aquaglyceroporins have a similar structure with a pore that opens up a little larger. And it's aquaglyceroporins, this is aquaglyceroporin 3. It's expressed in the basal levels of skin at a very high level. This is work of our friend Johan Ogren when he was a sabbatical worker in our lab. Now, it didn't take the beauty industry long to jump on this. I was visited in my laboratory several years ago by the executives from the Christian Dior company, who invited me to give a lecture in Paris. And of course, it seemed like they probably had a commercial explanation, and they did. They have a product which they would like you to purchase. Now, the beauty industry does not have the same responsibility as the pharmaceutical industry. This skin cream has some small substances which appear to lead to increased expression of aquaglyceroporin 3 in skin. You'll have to take their word for it. I'm not personally convinced, but the information was on this slide was on the back cover of one of the beauty magazines, and their advertisers had very bold interpretations. Visible hydration, spectacular results. And what's this at the bottom? The Nobel Prize in Chemistry. My mother, she's 96 years old. She lives in Minnesota. She's a farm girl. She never went to university, but when I showed her this, she smiled and said, "Peter, I think you're finally doing something useful." Our mothers know us better than we know ourselves, I think. I'm not sure if that was the most useful thing, but the presence of the aquaglyceroporins in fat tissue, shown on the left, will restore excess calories, and the aquaglyceroporin 9 in liver, shown on the right, work in tandem. We have a new disease in Western Europe and North America called obesity. Back when I was growing up, about 2 to 3% of children would suffer from obesity. Now, about 30% of children are obese and half are overweight. And the efficient storage of energy in the form of glycerol esterified into triglycerides in fat provides a store for when prolonged periods of fasting occur, and even in extreme exercise. The triglycerides are broken down, the fatty acids are released by fatty acid transporters, and the glycerol is released with aquaglyceroporin 7, where it's taken up by the liver and reconverted to glucose to maintain blood glucose levels, even at the end stages of starvation. And this is work of Jen Carberry, now a graduate student who's now down on the faculty at Duke University. The aquaglyceroporins, unfortunately, are permeated by arsenite. Arsenic is a toxin which we don't have problems with in the United States, but in Eastern India and Bangladesh, where the surface waters are contaminated with cholera, the groundwaters are tapped, but unfortunately many of those locations have very high levels of arsenic. So this study undertaken with Barry Rosen when he was at Wayne State shows the high permeability of the aquaglyceroporins to arsenite. So arsenite is uncharged at neutral pH, and by not having functional aquaglyceroporins in our liver and our kidney, arsenic excretion is diminished and toxicity accumulates. Now, in closing, I'm just going to talk very briefly about the aquaglyceroporin 3, which is present in red cells. It's not essential to have aquaglyceroporin 3 to have normal functioning red cells, but this protein has been seized upon by the malaria parasite to give it an advantage. When a malaria parasite enters a red cell, it will synthesize glycerolipids. And a single parasite will divide, double, up to 32 parasites, and kill the cell, eating the hemoglobin. And this is work undertaken with our colleagues Nirbi Kumar and Dominique Premineur. Nirbi is presently at George Washington University, and Dominique is at the NIH. So you can see in this micrograph of red cells that an individual red cell shown just left of center with a single parasite, and then to the far right, you can see a schizont where the parasites have divided to 32 daughter cells. And so this amplification, infection, lysis, reinfection, goes logarithmically. So a single parasite can lead to the appearance of 10 to the 10th parasites in just a couple of days, leading to horrific fevers and end organ damage. Malaria, of course, is passed by the Anopheles mosquito, and Anopheles also have aquaporins in their Malpighian tubules and their other tissues. We're working on the biology of that now. And alas, plants, as you would expect, are a rich source of aquaporins with up to 50 different aquaporin genes expressed in the rice genome. This is an Arabidopsis, and it's a very interesting example. This is prepared by my friend Rolf Kaldenhoff at the University of Würzburg in Germany. On the right, we see a parental Arabidopsis, and on the left, we have an Arabidopsis which has been engineered to downregulate the expression of the rootlet aquaporins. So it has only 10% expression. But notice the plant on the left compensates. The foliage appears normal because the rootlets amplify, each with reduced water permeability, but together compensating. So plants compete with each other for water. Now I have talked about the discovery and the permeation, the aquaglyceroporin aquaporin variants, structural models, and a number of the disease states where these aquaporins are involved. But it didn't prepare me for this. 18 years ago, the phone rang early in the morning, about 5:30 in the morning, October 8th. And on the other end, I heard a voice saying that this is an important telephone call from Stockholm for Professor Peter Agre. Are you Professor Agre? And I thought I knew what this meant. And I said, I sure am. And they shared that I would receive the Nobel Prize together with Roderick McKinnon for the discoveries related to channels. Rod studied the potassium channel and many other channels, and we studied the water channels. Now, the phone call went on, and I'm starting to imagine my family reacting to this, and my high school chemistry teacher, who was not impressed with my marks in chemistry, I must say, in high school. I straightened out in college. I imagined there would be some surprises. And of course, when I got to the laboratory in the morning, the young people were in their third bottle of champagne. The university president, Bill Brody, even came over to see me. And at that moment, I realized what this really all meant. Then I sprinted to my office, grabbed the phone, and the voicemail was filled with messages. And I came quickly upon one from our Norwegian colleagues. The voice was very, very familiar. And it said, Peter, we've just heard the news. It's unbelievable. And then a pause came, and the voice came back on. Peter, it's believable. Now, shown here are the young people in our lab at that day, and they actually originate from 8 different countries. It's a marvelous thing to have a research laboratory. But it's also marvelous to have family support. Here we were on the stage after the ceremonies, my wife, Mary, and our 4 children, and they really were wonderful. So the Nobel goes to an individual, 1, 2, or 3 scientists, but it really represents the work of the entire field. Because science is complex now. There are probably no investigators that individually on their own can do all of the work. So it really should be a reflection of the recognition of the transport field and the importance that that plays in health, and the potential importance, because we still don't have good ways to inhibit or stimulate the aquaporins. In closing, I'd just like to say that the joy of the discovery is one of the major drivers of science in my laboratory and other laboratories. But now I'm 72. I've closed my laboratory. I've been, for the last 14 years, running a malaria research program. And we have field activities in Africa. I'm delighted to say that in southern Zambia, where our main activity is located, the prevalence of malaria has been reduced by 95%. But it's still present in the north. There are big challenges. So the work, I feel, is important, but also the people we work with, the people in our laboratories, the trainees, our colleagues, our collaborators, and even our competitors. Together, I think we can move science forward, and we hope advance the public, the well-being of the public. So with that, I'd like to thank you. I apologize for the technical problem. I'm sure there's some cobweb in a computer somewhere, but I'm pleased to share our story with you, and I'll do my best to answer any questions you have. Thank you so much. Talking about water makes me thirsty.
Hi, Dr. Agre. Thank you so much for such a fascinating talk.
A few unexpected events, but
I think we pulled through nicely.
yes, I appreciate your calm demeanor.
Hello to everyone out there. Good evening. I am Charlotte Abney Solomon. I am the Associate Director of the Beckman Center for the History of Chemistry at the Science History Institute. And it's my honor to be with you here for the Q&A portion of tonight's program. So if you have a question for Dr. Agre, you can leave it here on YouTube in the chat, or you can email it to us at ullyot@sciencehistory.org, or you could send it to us on Twitter. We are @scihistory.org. So Dr. Agre, I have a question for you to start. I was absolutely fascinated as you were talking about the conditions that could arise from an aquaporin deficiency, or perhaps conditions that could be ameliorated by an aquaporin deficiency. So I was wondering whether and how it's possible these days for doctors to either induce or treat an aquaporin deficiency in order to treat these conditions.
Well, the inherited defects in the aquaporins are fairly uncommon. I think oftentimes the phenotype requires some extensive testing, because the function of these proteins appears to be most important at points of stress. The release of sweat, the ability to lyse triglycerides and release glycerol for gluconeogenesis, the ability to transport water in brain. Now, the aquaporin 4 in brain is at the location of the blood-brain barrier, and it seems to be the management of the edema in experimental animals shows a smaller degree of brain water uptake in the mutant animals. So if we could inhibit them, if we had a good inhibitor, that would potentially be a treatment to prevent brain edema. On the other hand, it could be possible then to trap fluid in the brain. So it's not yet been shown that there's a compound that is of clinical utility. I think there are a number of biotech firms. I was associated with one, but it appears to be a very difficult drug target. So I think the basic science of what they do in terms of physiology has been relatively straightforward. But to do something, as my mother would say, useful, is proving to be difficult. They're not always expressed and maintained at certain levels. They can be induced in some tissues. For example, chronic thirsting leads to an upregulation of aquaporin 2 in the renal collecting duct. When the person is rehydrated, then they release the excess aquaporin 2. So there may be some therapeutics, but we need some good inhibitors or some agonists. And again, they so far have not been successful. The brain aquaporin 4 has been shown to be directly linked to one form of brain autoimmunization. Neuromyelitis optica is a disorder. It's classified oftentimes with the muscular sclerosis patients, and the aquaporin 4 antibodies react presumably with epitopes in the aquaporin 4 in spinal cord and retina. So it can be used to diagnose that disorder, but it's not been yet useful to treat that disorder.
See, that's very interesting.
I'm hoping some of the young people will do experiments to show that these discoveries were actually more important than they are. Right now they're answers, but we don't know how to make the questions.
All right, so we've got a few questions that are coming in from the audience. The first one is actually very closely related to what you just said. Speaking of the young researchers, I really appreciated how you talked about the teamwork involved in science and how work like this is done by a lot of people together, and particularly young scientists in the laboratory. And so we have a question from the audience. What advice would you give to a young scientist just starting out in this field?
Well, that's a good question. There's a book written by Santiago Ramon y Cajal, Advice to a Young Investigator. And it's pretty severe. He basically says, have nothing else in your life but science. I think in the 21st century, the need for some work-life balance is important. But I think first off, an individual who has an interest in science, how things work, what makes them tick, is the curiosity that will spark the drive. there's a level of intellectual capacity and just the ability to get things done that are necessary. But I think the real interest in how things work, with the thought that we might then be able to make things better, is something that scientists all share. Making a discovery is a very heady event. I showed you Greg Preston, how jubilant he was, and we were all jubilant. But when something useful comes from that is the ultimate reward. So I don't think all young people are cut out for science, as certainly all young people are not cut out for musical careers. There's certain talents which seem to be innate, but I think the interest needs to be early in life. As children, we're all natural scientists. We love these observations. We call it magic, but in fact, magic with a scientific explanation is what draws our interest. I hope I provided some credibility with that answer.
I think so. So at the very end of your talk, you mentioned that you had moved on in your career from aquaporins to researching malaria. And we have a question from the audience that kind of bridges these two. They ask, would it be possible to selectively inhibit the aquaporin in malaria-bearing mosquitoes?
That's a tall request. We studied the aquaporins in the parasite, and the null parasites will invade red cells, but much less efficiently. In the end, when we did studies, I took the slides out to make it shorter. I could have left it in. Dominique Promeneur and Nurbi Kumar did these studies. They found that the malaria parasite, we were looking at Embergii, infects mice, normally will, after 7 days after injection of the parasites, the mice start to die, and by 2 weeks they're all dead. Those injected with the same number of parasites with the aquaporin gene targeted, they don't seem to show sickness until about 2 weeks after injection, but by 3 weeks the mice are dead. So we've prolonged their life, but this is a complete knockout. It doesn't seem like this is going to be a likely drug target. So while we are disappointed, there's plenty of other things to work on. And I think science is about learning and moving ahead. It would be nice if we had a new drug target, but I'm afraid I can't think it's going to be likely to turn out.
So another question from the audience kind of goes to that question on the other end. They ask, are genotyping diagnostics in routine use for any of the aquaporins? Is it something that can be at least diagnosed through looking at this?
Well, I think we will eventually be able to use genotypic information. Aquaporin 4, which probably is the most important clinically, is the one aquaporin for which no human mutations have ever been identified. We have mice with mutations in aquaporin 4. I talked about them a little bit. They look pretty normal, phenotypically normal. They were first engineered by Alan Verkman and his team out at UCSF. But when they're studied very carefully, it was found that they resisted brain edema. But the mice did have an increased vulnerability to epileptic seizures and more severe seizures. So it might be the mutations in the aquaporin 4 could be studied by genotypic analysis and sort out once and for all, because there must be a human counterpart, but so far it's not been established.
So another audience member asks, they say they have Sjogren's syndrome, which is an illness which features dry eyes and mouth, always thirst, and they're wondering, are aquaporins involved in this?
Yes, that's an excellent question. So Sjogren's syndrome has had 2 different reports in 2 different kindreds, identifying in one expression of the aquaporin 1 at high levels in the myoepithelium, and the other a mislocalization of the aquaporin 5. So it's not altogether clear that these results will have significance to a large number of other people. It's an autoimmune disorder, and it could well be an epitope in the Aquaporin 5, but I'm not confident that that's going to hold up for more than a handful of patients. Sjogren's syndrome is a significant disability, and as we get older and develop other medical problems, I for one have Parkinson's, and I need to take medicine for that, and it leads to increased dryness, and you become aware of what the people have to put up with. It's really sometimes quite uncomfortable, but usually not life-threatening, of course.
So another application question: would there be applications for aquaporin in water purification or desalination?
Excellent question. A number of engineering groups around the world have been at work on that. And there's one in Copenhagen. I have no financial stake in it. They are named Aquaporin LP, I think. And they have prepared cassettes for water purification. And they've been able to reconstitute the bacterial aquaporin, which is referred to as Aquaporin Z, Aquaporin Z. And these appear to be stable and lead to final purification of water. I'm sure this can be improved upon. And that would be very wonderful if we could have a system for desalinating seawater, which is a low energy machine. I think they've been working this in Denmark now for about 5 years, and things are looking promising. And again, they do have some commercial products available, but they're so far, I think, marketed only in China.
We have a more technical chemical question for you this time.
Well, these are the ones that I always get frightened of.
Well, this audience member asks, is the aquaporin water channel able to distinguish between the passage of D2O versus the normal H2O molecule?
That's an excellent question. It's technically challenging to measure it because we're not measuring ion conductance. We're precise measurements. We're measuring the rate of swelling or shrinking. The attempts to show a difference have failed to show a difference, but I'm suspicious. I suspect there is a difference.
So I have another question for you on the different kinds of aquaporins. You've taken us through a variety of different kinds, and I'm wondering whether various organisms vary in how many different types they have. Are some more essential than others? Is it just that each organism has a different combination? How do they vary from between different types of living things?
That's an excellent question. First off, why do we need 13 aquaporin genes? We probably don't need all 13 to survive, but if stressed, each has a special role that is probably not fully compensated by others. In terms of do all organisms have aquaporins? As far as we can tell, all wild-type organisms have at least one aquaporin, or one aquaporin and a aquaglyceroporin. But when we looked at laboratory strains of yeast, Saccharomyces, which are very viable in laboratories, turns out they have knockout mutations in their aquaporin genes that have been accumulated. So presumably these yeasts do not express functional water channels. And it's probably reflects the preparation of yeast when they're washed with fresh water. Those which are expressing aquaporins may actually lyse. So we know that yeast can live without them, but the wild type all have them. In terms of the human distribution, the kidney is the most complicated. I talked about the proximal nephron that have aquaporin 1 present, and humans with mutations in that look normal, feel normal, but they can't fully concentrate their urine. Those with mutations in aquaporin 2, which is just the final concentration of urine, and that's, of course, the intracellular water channel that moves to the surface of the collecting duct, they're severely affected. Those small children have to drink up to 20 liters of water per day to maintain hydration. And that there are also basolateral aquaporins in the collecting duct. Eye has, I think, 5 different aquaporins expressed, each in a precise location. So I think you can get along without it, but for that matter, you could get along without eyesight if you're living in an environment where everything is provided for you. I think we don't really know until the animals are stressed or the humans are stressed how valuable they are. Often I'm asked, are aquaporins permeated by gases? And Walter Boron, who was at Yale University, he's now at Case Western Reserve University, believes that they're permeated by carbon dioxide. And about half of the carbon dioxide in red cells must go through aquaporins, he believes. There are others that disagree. We've not worked on that ourselves. But if you're missing half of your capacity to transport carbon dioxide, it might be of no consequence unless you're being chased by a saber-toothed tiger, in which case you probably need all functioning gas channels to be operative to maintain or achieve the speed needed to evade the tiger.
That sounds like a very, very, very small scale analog to the condition of having maybe one kidney or one lung.
Yes.
You can live a happy life with just one, but once you're being chased by a tiger, you'd want both of them. Well, it looks like we have time for one more question. So I wanted to just ask you a little bit more about what inspired you to transition your research from aquaporins to malaria.
That's a very fair question. And in truth, Charlotte, my interest in global health preceded all of this. When I left Minnesota, a wonderful but cold northern state, the idea of working in the tropics and global health really caught my fancy. And when I was accepted at Johns Hopkins, I was so eager to attend because they have leading programs in global health. As a first-year medical student, I studied cholera, a horrible diarrheal disease. And of course, great progress has been made in cholera. The toxins have been purified, there are vaccines, but it was never clear how the water escaped from the diarrheal fluids. And of course, cholera victims can release gallons of fluid, go into shock, and die within an hour or just a few hours. So I was interested in global health. And when I started working on red blood cells, it was largely because my best friend and my lab partner, Van Bennett, my classmate from medical school, was doing frontier-breaking work on red cells. And of course, the most serious disease of red cells is malaria. So I worked on red cells initially thinking we'd get into malaria. But then when we discovered the aquaporins, we were in a situation we couldn't do everything. So if you can imagine you're out prospecting for gold and you find a vein of platinum, what do? You switch directions. But there was a point when I think we felt it's accomplished as much as we could in the aquaporins. We started looking at aquaporins and malaria. And then I became an administrative director responsible for field activities. And in my 60s and now in my 70s, I've enjoyed that immensely. I expect to step down as director of the Malaria Institute at Johns Hopkins. We have a great opportunity for a younger scientist. I'd like to pursue science diplomacy. So there's always something new, a new adventure in science. It keeps us excited.
What a fantastic topic, and what a fantastic trajectory. That's wonderful. Well, thank you so much again, Dr. Agre. It's been a real pleasure talking to you and a fantastic talk. I really appreciate it. And at this time, I would just welcome back David Cole to say a few words to close.
Well, Charlotte, thanks so much. And Dr. Agre, let me express our collective gratitude for a very stimulating talk, and most importantly, for your extraordinary commitment to your work. To our global audience tonight, we appreciate you joining us wherever you happen to be. Video of this program will be closed captioned and posted on YouTube in the coming days, so we invite you to revisit it and to share it. And on behalf of the Science History Institute, I wish you all a very good evening. Thank you.